Hello, my name is Macario Olmos, and my group was assigned the Fossil Atmosphere Zooniverse project. The purpose of our project is to look at microscopic images of ginkgo leaves taken from many points in time and identify two different types of leaf cells within them, the stomatal and the epidermal. The ratio of these two cells allows us to record how the atmosphere has changed over time, as more stomatal cells are what let leaves exchange gas. Because the leaves are very particular of how much CO2 it takes in, it has a different number of stomata depending on the concentration of CO2 in that time period. Since we have fossils of ginkgo leaves in from many different time periods, identifying the ratios will let us identify the concentration of CO2 in those time periods they originated from. I'm going to do a quick classification, that way we can get an idea of what that actually entails. So on our right, we have, we can mark stomas, regular cells, mark if we're not sure about something, and mark an unclear patch. Doing this, or actually I should first say what an example of each one is. So these big oval ones, which is identified here, they can look at just a few different ways, but they generally look like this. There's a few on here. So for this one, there's only 5. We only count what's inside of the square. So even though there's one right here, we don't count it. Now the rest of it is the actual time-consuming part. It is the regular cells, which there are a lot of. Anything that is enclosed within its own wall is a cell. So actually, even though I marked this one, I'm not 100% sure because it's hard to tell what's part of the actual base of it and what is a wall. So it kind of looks like there's one there, but just to be safe, not sure, because no reason to mess up the classification. Rather just have someone else look at it and either agree or disagree. So we're just going to work on that real quick. So marking the unsure part actually exemplifies one of the best reasons as to why it's better to have humans do it and have it kind of like aggregated over time. There's less error that way. If we have a computer do it instead, sure, you can train one to do it, and it'll get like better results every time, a higher positive mark. But it's better to have it done in groups because then you just have someone checking your work. That way, like for right here, I'm not sure if that's its own or not. And I'm not sure if this is a separate one or if this whole long area is one specific cell. So marking it allows someone else who is also doing stuff like this to look at it and either, yeah, no, that's a cell by itself, or that's a new one. Even right here, I'm not 100% sure, so I'll just mark it. Since it's group work, it gives me more confidence because that way I know that if I make a mistake, someone can be like, yeah, no, that's not right. That's completely wrong and not how to do it. So with all that said and done, I think this one's looking pretty good. See, that's another unsure spot because that could be a wall right there. That I'm not sure as well, so I'll mark that unsure. Thankfully, we didn't have to use the unclear patch. The unclear patch just gets a huge area, but we don't need that because we actually got everything done. This project's important. The climate is very sensitive to CO2 levels. And depending on it, it can have an effect on both economical and societal. It could have economical and societal repercussions because the CO2 can affect climate change. Yeah. CO2 can affect climate change, which affects the sea level, food production, storm strength, water availability, and a bunch of different things. But those are, the big ones. Being able to quantify the effects with a certain level of CO2 in a time period will allow us to make a projection of future environmental change. It won't be one-to-one, but it's better to have that so we can plan ahead and kind of get an idea of whether or not projects are working, if they're helping or if they're not helping, and just get a general range of it. With all that said and done, I'd like to talk about my personal experience working on this project. Just gonna mark that real quick. I'd like to talk about my experience with it, and then My classmates recorded their own little video that talks about their experience with it, and I'm going to put that after this. I think overall I had a fun time with doing these classifications. It was a little difficult at first. I remember in my journal entry I specifically stated, "Yeah, this is a little rough. This is kind of hard." But as the days went on, being able to pull on that memory made it a lot easier. You still had the days where some of it, like even in that one that we got compared to the earlier one, it's a lot fainter. The lines aren't as defined. This stoma is very defined, but the rest of the cells aren't. Having to deal with that, with some of it being really easy and the others being hard, made it very confusing at times. It was nice being able to contribute to a project, though, especially since this has an impact on environmental change, and it'll allow us to figure that out. I think the only thing I didn't enjoy about it, though, was by the end of our project, our week, or rather 8 days of having to do it, it felt really monotonous. But I think that had more to do with it being something we had to do for class, opposed to something that I am just volunteering for, something that I'm choosing to do. So I don't think it's related to that kind of distaste was related specifically to the project, but rather just that we had to work on it. I think besides that, this is a very fun project. I might even, depending on how other classes make their stuff look, maybe I'll just chip in and help out there. Especially because False Atmospheres is cool, but maybe there's other cool stuff. Thank you and for listening, and now I'm going to be playing my classmates' videos.
Hi guys, my name is Eve Araguz and I'm part of the Fossil Atmospheres group. Going back on the start of the project, I was completely confused, had no idea what to do. But once the professor went over how to start going into the Zooniverse website and kind of clicking and showing us, how to get to our classifications, I kind of got into the groove of it. Every day I did my 7-8 classifications, and at first I was still trying to figure out what a stoma was, and what if it's a tear, what if it's not a tear. So I was a little confused, but towards the end of the project I was able to identify most of the stoma and what was a tear, what was a regular cell. And it was pretty interesting. In the beginning, when I first started, I had compared the stomata to the Venus flytrap plant, just because of how it has the little hole in it, and it looks like it's just ready to chomp at you. And then looking at the cells, another thing that came to mind was it looked like a spider web. Some of them look like deep caves, or excuse me, caverns. And so it was a very interesting project. I've never done anything like this before, and was pretty pumped about it at the end. It makes me want to learn more. The thing that I enjoyed most was learning about it, and getting a better understanding of what I was looking at. Yeah, it was hard. Some of the parts were hard, like identifying what is really a stoma and what is an actual tear in the leaf. And there were some classifications that I looked at that were a lot lighter than others. It made me think that maybe, it was the microscope or whatever was, zoomed in too close or too far back, so it would change the coloring of the classification. I also noticed that there was quite a bit of debris on some of them, and again, that made me believe that the microscope was probably extremely close to the leaf when they were identifying, the stomata and and the cells. So if you've never done anything like this before, I highly suggest you take a look and learn something new. Learn how that affects looking into these leaves affects how how we're able to breathe better, or they use this to identify you know how to improve the atmosphere. So if you get a chance, take a look. It's on Zooniverse, and it's fossil atmospheres. Thanks.
Hi, my name's Ashley. I had the fossil atmosphere project. Was the task easier or hard? Well, during the beginning, I really didn't understand on how to find the stoma, and that was hard at the beginning. However, after my third visit, I began to understand and comprehend the project much better. Was the task interesting or not? I believe that the project was interesting for me. I enjoyed the different classifications and not just getting the same ones. Also facing the new challenges on how to find stomas. I believe that the project was interesting because it was intriguing because it wasn't just the same classifications. There was a variety. What did I enjoy the most and the least about this project? What I enjoyed the most was finding different classifications. Also, the purpose of the project I think was really cool and neat. For instance, we were learning about ginkgo trees and how they survived through 3 mass extinctions and finding them and solving them. I think it was just a really cool, this is a cool project, and the purpose was what made it was my favorite thing about the project. What I enjoyed, what I least enjoyed about the project was finding sometimes the images weren't as clear for the classifications, so that's what made it a little bit harder. But if I zoomed in and took my time, I began to understand the project much better. Did I encounter anything surprising, confusing? I really didn't encounter anything surprising. What was confusing was finding the stomas. As I said, that sometimes the classification in the image weren't as clear. But overall, it was a really great and interesting project. And
Hi, my name is Ferreira. I'm part of Group 1, and our title project is Fossil Atmospheres. I'm going to be talking a little bit about my individual experience and how I felt throughout the whole thing. One of the questions is if I felt that the task was easy or not. And for me, it felt like both. Sometimes it was easy, and sometimes it was hard. It was easy when classifying because I understood what I was doing. I knew what I had to classify. I knew what I had to do in the entries, but sometimes it was hard when I had to write the entries because it felt very repetitive and it felt like it was just going on and on and on, and I didn't want to write the same thing for every image, so that was the hard part. But we go to the next question, which is if the task was interesting or not. And I think that's what it made it interesting. It was that big challenge of having to come up with new things and looking at each image and understanding those, and each image was very, very different from each other, so that was interesting as well, to learn a lot about science and about the ginkgo leaves that we were classifying, all of that. So yeah, overall I enjoyed a lot this project. Besides it feeling a little bit repetitive, I learned a lot. I definitely did a lot of research and made sure to write that in my entries, and as a group we've been doing really great. I can't wait to see the final result of our video, so thank you.